So I'd like to thank Professor Samal for giving me this opportunity to present here. So today I'm going to talk about our journey to explore structure activity landscape of environmental chemicals and to identify activity cliffs. So the first question that I'm going to ask is, what are activity cliffs? So the activity of chemical can be quantified based on their binding affinity to a biological target. And if you have a bunch of chemicals which constitute chemical space, and you take all the pairs of chemicals and compute their activity difference, and that can be visualized on something called structure activity landscape. Now if you have chemicals which are very similar but activity wise they are very different, so that particular chemical pair can correspond to a sharp peak in the structure activity landscape, which is basically called activity cliff. Now what is so important about activity cliffs and why should we care about activity cliffs? So that answer is given by this editorial which is published in 2006 by Professor G. Majiora. So this editorial talks about the fact that activity cliffs are very much inherent in chemical data sets, and if you are building QSR models, this QSR model relies on similarity principle, which suggests that similar chemicals will have similar property, and if your training data set has activity cliffs, then that model prediction are going to be flawed because activity cliffs do not obey the similarity principle. Now the question comes then, how to identify activity cliffs? So this particular answer has been given in chemical space which is relevant for drug discovery research, but then the question comes, how about environmental chemical space, whether this kind of activity cliffs are being characterized or not. So we are actually addressing this particular question, how to identify and characterize and analyze activity cliffs in environmental chemical space. Now we have done 3 pieces of work, so I'm going to talk about the first part where we have analyzed and explored the activity cliffs in androgen receptor binding chemicals. So how to identify activity cliffs? So this is the structure activity landscape where the peaks corresponds to activity cliffs. So this landscape is not so informative, but if you go to a 2-dimensional space which is called structure activity similarity map, and the x-axis corresponds to the chemical similarity between all the pairs of chemicals, and the y-axis is the activity difference for all the pairs of chemicals, and with some threshold of chemical similarity and activity difference, one can actually have 4 different quadrants, and the quadrants that is important for us is the 3rd quadrant. So all the chemical pairs which falls into that particular quadrant, they are highly similar, but the activity difference wise they have large difference in activity, which is called activity cliffs, and we are particularly focusing on this particular quadrant. Now what we have done in this work is that we have curated a list of 144 androgen receptor binding chemicals with their relative binding affinity, which is reported in this particular literature, and we have employed this SAS map approach, and for all the pairs of chemicals among 144 androgen receptor binders, and we have focused on this 3rd quadrant which basically gives the activity cliffs, and we identified 86 such activity cliffs which is formed by 41 chemicals. And we then asked the question, are there any chemicals which forms more number of activity cliffs, which is basically called activity cliff generators, and we were able to find out 14 such activity cliff generators. So the next obvious question that we are going to ask is that, are there any structural features which are behind the formation of activity cliffs? So what are the structural features one can consider is that for a chemical one can take the scaffold, which is a core structural framework, and one can also take the R groups which are basically connected to the scaffold, and one can also consider the position at which these R groups are connected. So using these structural features we have a workflow by which we are able to classify these activity cliffs in different categories. So to give you example, so these 2 chemical form activity cliffs, the only difference here is the position of this OH group. So since the position is different, that is called, the topology wise this chemical pair differ, that is why it is called topology cliff. So with this I am going to go to the second part of my talk, where we are asking a much more deeper question, that is how one would take this approach to a much larger data set which is more practical for people to use for building models, and we are also asking some more interesting question in this. So, so the first question that we are asking here is that, are there any larger data set which is a benchmark data set for people to, well, people building QSR model, they are likely to use that? The answer is yes, and the library is ToxCast library, where, so ToxCast is a US EPA project where they have used high throughput assays to screen nearly 10,000 environmental chemicals. Now, what we are doing here is that we are collected the data set of, for a particular receptor, that is thyroid stimulating hormone receptor, from ToxCast library. And what is the data that we have collected? That is, so we have collected the chemicals which are reported in agonist assay, that is chemicals which are tested for whether they can have similar function like natural hormones, and we have also collected chemicals for antagonist assay, that is chemicals which can inhibit the receptor. And for these chemicals we have compiled the activity value reported in ToxCast, and also the mechanism of action annotation, that is whether this particular chemical is active in agonist assay, active in antagonist assay, and so on. So with this, first obvious thing that we did is we used this SAS map approach and identified activity clips both for agonist and antagonist data set, and also activity clip generators. The next thing that we are asking is that, are there any approach which do not depend upon the chemical fingerprint? So in the SAS map that I showed here, the Tanimoto coefficient one compute using chemical fingerprints. Now we are asking, are there any approach where we can consider the similarity between chemicals but not with fingerprints? So the answer is matched molecular pair approach, where if you take two chemical, and if these two chemicals change at a single site, you call them as a MMP. So how to identify a chemical which satisfies MMP properties? Now you take two chemical and you fragment this chemical into all different pieces, and you see which fragment of this is common between the two chemicals. So in this case the left and right sides are common, which is constant part, and the middle fragments are transformed. That is, if you are going from first chemical to second chemical, this is the only change which is happening. Now since we are looking for activity clips which is basically high similar chemicals, so we are imposing certain size restriction and also activity difference threshold, and we are saying that if a chemical pair satisfies this condition, then this forms a MMP clip. Now this is an example of MMP clip where this chemical and this chemical are same, the only difference is in this middle region. And what we observe is that the MMP clip that we have identified in the agonist and antagonist data set, they are always a subset of the clips that we identified from the SAS map approach. So the next question that we are asking is, similar to analyzing structure activity landscape, can we analyze the structure mechanism relationship? That is, you have similar chemicals but the mechanism wise they differ. So what we have done here is that we have taken the common chemicals between agonist and antagonist data sets, and then we are asking a question, which are the chemicals are similar. Then if one chemical is active in agonist assay and another chemical is active in antagonist assay. So this is the workflow by which we are able to classify the chemicals in different categories based on their mechanisms, and this is some examples where so these two chemicals are strong MOA clips, where the first one is active in antagonist assay, whereas the second chemical is active in agonist assay. So these are the number of clips that we are able to find out from this data set. Now with this I will be going to the third part of my talk. So here we are asking the question, how to analyze the multi-target nature of the structure activity landscape of environmental chemical space. So in general, chemicals can show promiscuity in binding to multiple receptors, and this particular notion is very well studied and analyzed in the context of pharmacology, and the same concept is not very well studied in environmental chemical space. So however, there are some evidence, for example bisphenol A, which actually can bind to multiple endocrine receptors in humans, and in this context ToxCast has again screened several thousands of environmental chemicals across all different human endocrine targets. So what we have done is we have focused on 8 human endocrine receptors, and we have collected again the corresponding agonist and antagonist data sets, and here we are asking this question that can we find chemicals which are similar, but their activity wise they are very different with respect to one target, that is single target cliff, and then with respect to 2 target and 3 target correspondingly. So to answer that particular question, we are using DAD map and TAD map to analyze multi-target nature of activity landscape of these environmental chemicals. So what is DAD map? So DAD map is a 2D map basically, and here how do you construct a DAD map? So for receptor T1 and T2, first take the common chemicals, and then you take the chemicals that are similar between these common chemicals, and then the x-axis, then you can construct the DAD map for these common chemicals with respect to T1 and T2 receptor, and the axis of the DAD map actually corresponds to the difference in activity for with respect to receptor T1 and T2, and with a threshold of activity difference small t, one can divide this DAD map into 5 zones, and the zone 1 and 2 will give you dual target cliffs, and whereas zone 3 and 4 will give you the single target cliffs, and zone 5 will give you the continuous R where there is no cliff. Similarly, one can also extend this approach to DAD map where you have 3 receptor. So DAD map is nothing but the combination of 3 DAD maps, and if a chemical pair forms activity cliffs with respect to all the 3 DAD maps, that will be basically triple target cliffs. So here this is so what we have done is we have 8 receptors, so we have taken all the dual target combination, that is 28 dual target combination and 56 triple target combinations, and for all these combinations we have constructed the DAD maps and TAD maps. So this is one such example I am showing where these 2 chemical form dual target cliffs with respect to these 2 receptor, and this is a TAD map where this gives triple target cliff, and these 2 chemical forms triple target cliffs with respect to these 3 receptors. We have also used MMP based approach to identify activity cliffs for all possible combinations of targets, and what we observe is that MMP based cliffs are always a subset of activity cliffs which we identify from DAD and TAD maps. We have also classified the activity cliffs into different categories for all the different combinations, and we observe that the R group cliff, that is activity cliffs where you have scaffolds are similar but only the side chains, that is the R groups, are different, that is the maximum number of cliffs one can identify among all the activity cliffs. So in summary, so we have considered different kind of environmental chemical spaces, and we have analyzed their structure activity landscape and identified activity cliffs. We have also used MMP based approach to also identify activity cliffs. We have also analyzed the structure mechanism relationship and identified different MOA based cliffs, and we have also analyzed the multi-target nature of chemicals, environmental chemical space, and identified multi-target cliffs. So we hope that these insights will aid in the development of better toxicity predictors which will help in characterizing human chemical exposure. And these are the references where the first 2 papers are published and the last one is in bioarchive. And I would like to acknowledge my advisor and also my co-authors. Thank you.
Thanks for staying in time.
So this is more of a speculative question. So you are trying to identify specific activity cliffs for given chemicals.
Yes.
So if you take into consideration the structure or certain features of the pocket of binding of these things, do you think that you will make a much better increase in terms of prediction of what is happening?
Yes. So there is a paper by Medina Franco, who is a very well-known person. So there is a paper titled Interaction Cliff. So it sort of talks about how protein in binding site interact with the different chemicals, and based on that it actually defines different kind of cliffs. But we are not going into that direction because we need the co-crystallized structure for that. Then only we can analyze that. So
here, yeah. So it's a very interesting approach, but as a follow-up question, as it were, as you know, in GPCRs, the agonist binding site is distinct from the antagonist binding site. So how would you take into account that the binding site itself may be different?
So, ma'am, so we have only considered whatever data set is given by the toxcast. So, toxcast actually tells which one is agonist, which one is antagonist. So, we have not gone into that area. We are just taking the binding affinity values which is given, and based on that we are analyzing.
Right. I guess you're using such data sets where the binding site is coincident. Okay.
Yeah, I mean, yeah.
Right. Okay.
Questions, so let's thank Ajay again.